Hello everyone, welcome back to my channel X Gene and Protein X. In this tutorial video, I'm going to talk about selection of restriction enzymes and ligation using T4 DNA ligase. So let's begin. So first we need to know that how to select restriction enzymes. So let's say that here is the vector and here four different restriction enzymes. So if I want to insert my DNA fragment here, then I need to choose these two restriction enzymes that I pointed out. And if I want to move this fragment here, then obviously the restriction enzyme should be the different, and here is the pointed out these two restriction enzymes should be selected. And again, if I moved in another location, then obviously these two restriction enzymes should be selected for this inserted DNA. So always remember that when you insert the fragment, then the beginning and the end of that fragment, that restriction enzymes need to be chosen. So here is the another example. If you see here, then it will be more clear. So here is the AGR2 fragment I inserted here. So for that, the XBA1 and NOT1, these two restriction enzymes I selected. But if I move this fragment a little bit downstream, then obviously two different restriction enzymes I need to choose. There is the NOT1 and BAMH1. Okay. So next, after selecting the right restriction enzymes, you need to digest your fragment as well as your vector with the restriction enzymes. So as a result, you will get the two sticky ends in the fragment as well as your vector. So as you see, this is it pointed out here. So here is the red color in the both the 5' and the 3' end. So here I will get the sticky end for the fragment, and as well as simultaneously I will get the sticky end in the 3' and 5' position for the vector as well. So the violet color, it indicating the sticky end for the vector. Alright. So now is the pre-ligation steps, that is the restriction digestion. So how to digest the fragment and the vector with the restriction enzymes. So here is the insert DNA fragment. So for the insert DNA fragment and for the vector, you need to prepare the reaction mixture simultaneously to different tube. So what you need to prepare here, two different restriction enzymes that you need to be selected. So that two restriction enzymes, buffer, and for inserted DNA, so inserted DNA you need to select here, and also for the vector DNA in the another tube for the vector DNA you need to include here, and additionally the DD water up to 50 microliter. So for the insert DNA, around 0.5 to 1 microgram is enough, and for the vector DNA usually it is 1 microgram. Then when you prepare all of this mixture, this reaction mixture, then it need to be incubate 1 hour at 37 degree temperature. So after digestion, you will get the digested vector and as well as the digested fragment. So next, what you need to do, so after getting the digested fragment, then you need to purify because it contains the enzymes and other impurities. So you need to get the purified DNA fragment. So usually we do the gel purification. So if the concentration is 1 microgram or more, then you need to choose for the gel purification. But if the concentration is less than 1 microgram, then obviously you should not go for the gel purification, then you will not get enough concentration. So in that case, you need to directly use the column purification. So after purification step, you will get the purified insert DNA fragment. And similarly, for the vector, also you need to, the digested vector, also you need to purify this digested vector to get the purified vector DNA. So in this case, normally for the 1 microgram, also you need to do the gel purification and then you will get the purified vector DNA. So when you have the purified insert DNA fragment and also you have the purified vector DNA, then this is ready for the ligation. So we need to go for the next step. So next is the ligation using the T4 DNA ligase. So this is the conventional method that we normally use, the T4 DNA ligase. So here is the, as I said before, that for the vector and for the insert DNA fragment, in both cases you will get the sticky end. So here the insert DNA fragment, it will be inserted to the vector DNA and then it will join together and you will get the complete plasmid with the inserted fragment. So usually this is the reaction mixture you need to prepare. So digested vector, usually the 25 nanogram, digested insert DNA, that is 125 nanogram, and along with that T4 DNA ligase 10x buffer and DD water up to 10 microliter. But the manufacturer, you need to follow the manufacturer instruction. So according the manufacturer instruction, you will get I think the digested vector and the DNA fragment, the ratio should be the different, but usually it is the 1:5. So of this reaction mixture, so you need to incubate at room temperature for 1 hour. Then, so this ligated product you need to directly transform to the 50 microliter DH5 alpha with the 5 microliter ligated product. So next is the working principle of the T4 DNA ligase, how it works. So usually the insert DNA, it has the 3' hydroxyl group, and for the vector DNA it has the 5' phosphate group. So the T4 DNA ligase, it catalyzes to form the phosphodiester linkage. So this is how this T4 DNA ligase, it ligate the insert DNA with the vector DNA. So this is all about this ligation and also the restriction enzyme selection. So I hope this video will be helpful. So if you like this video, kindly hit the like button, share it, and if you have any queries or if you are not familiar with some kind of steps, then you can request me for that video, so I'll make that video on it. So thanks for watching.